Today's webinar, as you know, will be focused on CryoEM in the world of drug discovery, with a particular focus on the computational aspects of the technique. I'll start by giving an introduction to the CryoEM method, which of course may be reviewed for some of the audience. Next, Jakob will explain why CryoEM is useful for drug discovery and what role it is expected to play. I will then get into a bit of detail about the computational phase of 3D structure determination, highlighting the key concepts, pitfalls, etc. Jakob will provide some examples and details about how these tools are already being exploited in drug discovery today, and I'll end by talking about some of the exciting advancements that are happening now, as well as ideas for the future. Let's start with some very high-level context. Inside of all organisms, inside of every cell, there are very small but highly intricate biomolecules called proteins, which carry out every biological process of life. That includes everything from neurons firing to muscles activating to the metabolism of food. Tens of thousands of different types of protein molecules work together to make us alive. A drug molecule is a small molecule that can bind into a specific site within a target protein in order to modify that protein's function. A correctly shaped drug molecule binding with the right protein in just the right way can cure a disease or treat a condition, and the goal of drug discovery is to find those small molecules that are safe but effective. Intuitively then, it's clear that the three-dimensional structure of a target protein molecule is a critical source of insight for drug discovery, which shows not only how a protein actually works, but also geometrically how a drug might interact with it. Unfortunately, most important protein molecules in our cells have been very difficult to manipulate or study, and solving the 3D structure of many of them has been an impossibility. That is all changing thanks to a new structural biology technique, CryoEM. CryoEM, or cryo-electron microscopy, is a recently developed, very powerful technique for 3D structure determination of individual protein molecules. CryoEM is an imaging technique that works on a physical liquid sample of a protein molecule target by taking pictures, then computationally reconstructing the 3D structure of the target. It's fair to say that today CryoEM is revolutionizing structural biology, providing insights where previous techniques could not. The concept of CryoEM was initially conceived decades ago, and at the time was not quite feasible in real life, but through a very strong effort of the academic community and technology development on various fronts, CryoEM underwent a resolution revolution around 2014, and it has since been possible to obtain high-resolution 3D structures of very important proteins. A large global community now drives progress on all fronts, and the pioneering work in CryoEM was awarded the Nobel Prize in 2017. Today, the entire technique is mature enough to be scaling up in usage rapidly, but still many more developments are on the horizon to push the envelope, expand the scope, and make the entire technique robust. CryoEM is already widely used by thousands of labs globally in the academic setting, and industry adoption is blossoming. Some groups and companies like NVIDIA, Merck, and Structura, the presenters of today's webinar, are working at that frontier to develop CryoEM into a tool that can be used as a critical driver of structural insights for drug discovery projects. There are three main phases in a CryoEM project: preparing a sample, imaging the sample in 2 dimensions, and computationally curating, sorting, reconstructing, and recovering high-resolution 3D information about the target from the raw image data. The computational recovery of structural information and how it can be useful for drug discovery is the focus of today's webinar, but it is critical to understand the sample preparation and imaging phases which give rise to the challenges that must be overcome computationally. Preparing a protein sample for CryoEM involves purifying the protein molecule of interest, applying a thin film of liquid sample onto a sample grid, and rapidly plunge freezing the grid to vitrify and preserve the protein in a near native state. The result of this preparation is a grid, which contains thousands of small, typically 1-micron holes, each containing a frozen thin film of ice, inside of which are embedded millions of individual protein molecules, all frozen in random orientations. A sample like this has a few interesting characteristics that must be considered. The quality and thickness of ice is paramount. Thin ice is ideal. The concentration of particles must be sufficient to produce populated images. Heterogeneity in the sample, caused either by imperfect purification or by the existence of multiple conformations of the target, is a very common occurrence. The random distribution of orientations of the particles is also critical. Having all the proteins face one way is not ideal. And aggregation and denaturation of the target can also frequently occur and can be an issue. Once a sample is created, it is placed into a very powerful, extremely high magnification transmission electron microscope, or TEM. The TEM is able to take pictures of the holes in the sample, and in those images, individual proteins can be identified. Due to the delicacy of biological molecules, the electron dose must be kept very low in the microscope, meaning the captured images have a very high noise level. It's like taking a picture at night with the flash off. In each image, the microscope captures a 2D projection of the proteins in the ice. Data collection in CryoEM is done in phase contrast mode, meaning that each image is incomplete due to electron interference. Microscope aberrations, stage drift, and sample deformation during the imaging process can reduce the quality of captured images. And radiation damage, as I mentioned, must be minimized, but it still occurs, meaning that the same patch of ice can only be imaged once. And in each image, the signal-to-noise ratio can be as low as 1 in 100. These characteristics of the data mean that huge amounts of data must be collected and passed on to the computational stage in order for 3D reconstruction to be possible. It's typical to collect several thousand micrographs containing several million particles for a single target in CryoEM. The computational phase of CryoEM is also a multi-step process. First, raw data is pre-processed to estimate and correct for motion, sample deformation, and microscope optics. Next, in each image, the individual particles of protein must be detected and located. Millions of particles may result from this process, with many being junk, broken, or contaminants. Sorting, clustering, and curating those particles in 2 dimensions allows for the removal of most junk, and the remaining particles are then used for 2D to 3D reconstruction. We will discuss this core part of CryoEM in more detail later. Heterogeneity can also be resolved at this stage, and the best particles can be used for high-resolution refinement in order to recover ideally near-atomic resolution details of the target protein's 3D structure. These steps constitute the single-particle CryoEM workflow. And now that we've got a grasp on how CryoEM works, Jakob will explain why it's such an interesting and exciting prospect for drug discovery.
Thank you, Ali. So as Ali has explained, in the last few years, CryoEM has become interesting for the field that is called structure-based drug discovery due to the resolution revolution. The classic approximation to this technique, structure-based drug discovery, is giving a protein that we call the target related to a disease and a molecule that alleviates that disease by making some effect on that protein, and a molecule, we call it a compound or a lead compound. Structure-based drug discovery is based on the study of how the compound and the target interact at an atomic level under the design of that compound to make it a useful drug. That is usually done by solving the atomic structure of the protein in complex with a molecule, and then studying how the compounds should be modified to interact in a better way with the target, with the protein, increasing their potency, selectivity, solubility, or other characteristics. This has been done for years by techniques like X-ray crystallography or NMR, but now CryoEM offers the possibility of studying some targets that have been out of reach for these 2 techniques because of their size, conformation and heterogeneity, or flexibility, or because they are very difficult to express or purify, and therefore they are produced in very small quantities. This technique, CryoEM, besides that, offers the possibility to study the proteins in their native state, and also of working with membrane proteins, specimens that are usually complicated to solve by other techniques, but that constitute the targets for over 50% of all modern medicinal drugs. So the fact is that a few years ago, CryoEM didn't have a resolution good enough to be used for drug discovery, but that has changed dramatically in the last few years. That can be seen by a steady growth in the number of structures solved by CryoEM in the literature. So if you pay attention to the graph at the right, you can see how the number of structures publicly deposited in public repositories have been increased in the last few years dramatically, and they continuously increase. They are still continuously increasing. But not only that, also the level of resolution, that is a measurement of the quality of the structures, has improved dramatically in the last few years too. But what is resolution? So resolution is, in structural chemistry, is the distance in a map at which 2 points can be distinguished. Let's say you have a cryodensity map with resolution of 3 angstroms. Then if 2 points of that density are closer than 3 angstroms in distance, they cannot be distinguished as 2 individual points. But if they are separated beyond that number, they can. Here you have an example with tyrosine. At 4 angstroms, you have a poorly defined shape for the side chain of that residue. So CryoEM provides the mesh, the blue mesh that you see, and the sticks are the model that we build based on that mesh. At 3 angstroms, you can distinguish the global shape of the ring, but below 2 angstroms, you start distinguishing the hole in the aromatic group. That's because the distance between the 6 carbon atoms in the ring, that is around 1.4 angstroms, and then make a hole around 2.8 angstroms in the center. So when you have a resolution better than 2.8 angstroms, you can see that hole. Depending on the question that you have for drug discovery, resolutions around 4 or 5 start being usable. For other questions, you will need better. When CryoEM entered that range of resolutions, it started then being useful for drug discovery. At the right, you have a detail of a publicly available CryoEM density map of the proteasome. What CryoEM provides is called a 3D electron density map of the protein that we represent usually as a 3D isosurface. So the blue density, the blue cloud that you see is the electron density map. It's a section of the proteasome. That map can be used for a variety of things depending on the resolution. So now I'm showing you the model that we create based on that cloud, on that electron density map. If your resolution is close to 2 angstroms, you can easily distinguish the side chains of the amino acids. It is important to notice that the value of resolution is an average through the whole map. Some residues will have a slightly better resolution, some slightly worse. So I would focus on these 2 bilines to see well, I don't know if you see my pointer. I would focus on those 2 to see the differences. If you decrease the resolution from 2 to 3 or 4, you start losing details. Depending on the resolution that you have, then you can use the map for different things, from building the model ab initio at the highest resolution and be able to see all the atom interactions between the compound and the protein in the binding pocket, to use modeling or molecular dynamics, fitting, or understanding the mechanism of action of the protein with lower resolution levels. But coming back to drug discovery, this figure represents the different stages in the development of a drug in which CryoEM can be involved from a structural point of view before the stages of preclinical and clinical trials. The figure has been extracted from a paper published by my colleague, Giovanna Scapin, which I will mention a few times during this talk because of her contribution to structure-based drug discovery and her research done in the insulin receptor by CryoEM. In the first stage, target identification, the determination of a single structure of the target may be enough to identify the target binding site and characterize its mechanism of action. During the hit identification stage, structure determination is used to solve the structure of the target in complex with different lead compounds that have been identified by other techniques as promising drugs. Once the structure has been solved, an iterative cycle or redesign of those molecules testing on structure determination may be necessary to optimize the different compounds and achieve the desired pharmacology, solubility, potency, selectivity, etc. From target identification to lead optimization, the number of structures that may be necessary range from 1 in the target identification stage to hundreds in certain cases in the lead optimization phase. So just to give you some examples of important targets that have been solved by CryoEM in the last few years, you have the structure of the Zika virus or the structure of the HIV strand complex intasome at the bottom left. That is the molecule of the HIV virus responsible of introducing the viral DNA inside the cells. At the bottom right, you have the gamma secretase. It's a membrane protein, quite small, 170 kilodaltons for a CryoEM specimen. It was solved 4 years ago, and at that moment, this structure was a revolution in the field due to the level of resolution for a membrane protein that was also so small for the CryoEM standards. The dysfunction of this protein causes the accumulation of beta-amyloid peptide that is believed to be one of the causes of Alzheimer's. In the top right, you have the human glucagon peptide 1 receptor in complex with a peptide and a G protein heteroprimer. This is what is called a GPCR, a G protein coupled receptor. This type of protein is a member of a specific family of membrane proteins that are targets of 30% of the drugs in the market. They are, in general, very challenging to work with because they are very difficult to purify because they are not very stable. In this case, the whole complex is 150 kilodaltons, and there have been very important advances in the study of this kind of proteins by CryoEM. In this specific case, their structure could be useful to fight type 2 diabetes and obesity. And with this, I leave the presentation to Ali.
Thanks, Jakob. Jakob has now given us a clear idea of how CryoEM fits into drug discovery. In order for drug discovery processes to use CryoEM in the inner loop for target identification, hit identification, and especially lead optimization, all parts of the CryoEM workflow need to be made as robust as possible. This is what will allow large pharma drug teams, smaller biotechs, and academic labs working on disease indications to treat CryoEM as a powerful but black box technique that can shed light wherever needed in a drug project. The computational phase of CryoEM is the one that converts reams of 2D image data into actionable insight, as Jakob mentioned, in the form of 3D structures. It is where all the details and challenges from sample preparation and imaging must be dealt with, and as such, the tools, algorithms, and software used must be able to deal with a vast array of different sample conditions, imaging conditions, molecular shapes and sizes, heterogeneity, disorder, etc., that are present in real valuable targets. The field of CryoEM has been fortunate to have many dedicated people working on it for decades to bring us to the point we are at today. Our team at Structura is building the CryoSpark software package on that foundation by developing new advanced techniques and optimizing existing methods with the goal of providing academic scientists and industry organizations with powerful user workflows, extremely efficient processing based on new algorithms and NVIDIA GPUs, and algorithms specifically designed for the drug discovery use case, which can help extract as much valuable structural information as possible. Now, going into a little more technical detail, I will explain some of the intricacies of this computational phase of CryoEM that we work on. Reconstructing a 3D structure from 2D image data in any context is a difficult problem. In CryoEM, it is made even more difficult by the characteristics of the data that we covered earlier. Notably, in each experimental protein image, the pose of the molecule itself is unknown. We don't know which way the molecule is facing. The noise level is very high, so each image contains very little information individually, and the noise cannot be reduced without causing specimen damage. Typical datasets contain millions of images that must be processed, and each image is actually only a partial observation due to the microscope's contrast transfer function, or CTF, which represents the destructive interference of electrons within the microscope. On top of all of that, each particle observed is not necessarily an image of the intended target. Heterogeneity in a sample can result if there are contaminants or multiple conformations of the target molecule. At first, this problem seems daunting, but in fact, it is a common type of computational problem that occurs across the disciplines of computational statistics and machine learning called a large-scale inference problem. The key reason it's actually possible to solve is that we know how a microscope works. If we knew a 3D structure, we could predict what images would be generated by a microscope using physics. So the approach to solving a structure is to try and invert this connection. We want to ask, what is the 3D structure that best explains the data we have observed? The algorithm that can actually be used to answer this question is known as iterative refinement. I'm going to explain this algorithm in detail because for anyone who will be using CryoEM for any purpose, understanding this algorithm and its characteristics provides all the insight for why results might be good or bad, where things can go wrong, and where there are areas for improvement. The algorithm involves two steps. The first step, alignment, starts with an initial guess of the 3D structure of a molecule. And then, for every single image in the dataset, tries to find an orientation from which the 3D structure best aligns with the experimental image. This must be repeated for every image in the dataset. In the second step, all the particles, along with the updated alignments from the previous step, are used to reconstruct a slightly better version of the 3D structure. This is called reconstruction or back projection, and is actually identical to how a CT scan of a person will be pieced together in 3D. These two steps are iterated, and the result is a high-resolution refined structure. The more general name for this algorithm in statistics is expectation maximization. You may often hear people talk about maximum likelihood methods or Bayesian methods for cryo-EM structure determination. In fact, these are all closely related in that they are all simplifications of various degrees of the expectation maximization algorithm that is a staple in statistical inference theory. Expectation maximization is guaranteed to converge to a solution if you repeat the two steps. However, a major issue is that if the initial guess was wrong, the algorithm can easily converge to the wrong solution. You can also see clearly how this kind of an algorithm would require huge amounts of computation. In each iteration, we need to independently look at every single image, compare it to every possible view of the 3D structure, and find the viewing directions that are plausible. Finally, a question arises of when to stop iterating. and how much to trust the result of this kind of an algorithm. Validation is crucial for being able to interpret CryoEM results. The need for a correct initial guess of a 3D structure in cryo-EM refinement has previously been a bottleneck in the field, where it has been difficult to work on proteins for which the structure is not already known. The reason that ab initio reconstruction, meaning reconstruction from scratch, is difficult is that the cryo-EM problem is non-convex. We are searching in the space of 3D structures for the one structure that best explains the data, having the highest likelihood. Starting from an incorrect initialization, the iterative refinement algorithm would climb the nearest hill and yield an incorrect result. Luckily, it turns out that this problem is solvable with a special type of algorithm, first developed by the Structura team and implemented in CryoSpark. The algorithm, a type of stochastic optimization, can effectively explore the space of 3D structures while optimizing the likelihood, and recover the correct 3D structure from a randomly generated incorrect initialization. This is key because it allows cryo-EM to be successfully used without human bias and without prior structural knowledge on new classes of proteins. This video shows how ab initio reconstruction can be performed on 3 very different protein molecules. The algorithm is sufficiently robust to be able to recover symmetric structures like the CryoEM on the left, membrane proteins like the ATPase in the middle, and large complexes like the ADS ribosome on the right, all without any prior structural knowledge or retuning. Ab initio reconstruction can also play a role in resolving heterogeneity in cryo-EM datasets. In this case, multiple independent random initializations can be used, and the algorithm can simultaneously classify and reconstruct from scratch multiple different states of a protein target that are present in the data. In this case, a single dataset of 150,000 particles was used to recover 3 different conformational states of the ATPase molecule. And looking closely, you can see that in the 3 states, the central stalk is in 3 different rotary positions. The robustness of this algorithm in CryoSpark makes it possible to work on all kinds of samples without knowing what they contain beforehand, and is critical to turning cryo-EM into a black box tool. Once ab initio reconstruction yields a correct initial model, we can return to the iterative refinement algorithm to recover high-resolution detail of a target protein. Iterative refinement, as we discussed, is computationally expensive due to the requirement to align all particles to a 3D reference at each iteration. This used to be a major bottleneck for recovering cryo-EM structures, and simpler naive algorithms still require massive amounts of computational resource and CPU or GPU time, and sometimes the total wait time for a user is measured in days, especially on larger or more complex projects involving millions of particles and heterogeneity. It turns out that a huge reduction in the computational expense of iterative refinement is possible with a more advanced algorithm called branch and bound in CryoSpark. Today, for example, this dataset of 100,000 ribosome particles can be refined to 3.2 angstrom resolution in just 11 minutes on a single NVIDIA GPU in a workstation. These dramatic speedups lay the foundation for making cryo-EM a scalable technique in the drug discovery workflow. Fast refinements are particularly important because real cryo-EM workflows today require many repetitions of refinement with different subsets of particles to reach maximal resolution. The third issue with the expectation maximization algorithm is that repeating iterations can cause noise from the experimental images to build up in the reconstructed structure, which then affects alignments in the next iteration, and eventually can yield incorrect high-resolution detail in the final result that is not actually present in the data. This problem, known as overfitting, is a common and very deep concept in statistical inference generally. In cryo-EM, overfitting and estimating the resolution of a reconstruction are closely related and have been somewhat hotly debated in the past, considered by some to still be open areas of development. Currently, one standard tool is the gold standard Fourier shell correlation, which provides a reasonably effective and objective measure of the reconstruction quality of a refinement. Measures like this are critical for real-life application in drug discovery, where decisions involving years and large investments may hinge on small details at near atomic resolution. The gold standard Fourier shell correlation, or FSC, works by splitting a dataset into 2 independent halves, which are separately refined. The reconstruction from these halves are then compared in the spatial resolution domain, and signal at different resolutions from the 2 halves are compared. Where the two halves agree, we have high confidence in the structure. The trustable resolution of the refinement is limited to resolutions where the two halves agree. In this way, today's high-resolution CryoEM refinements can be statistically validated and trusted for decision-making in a project. Now that we've covered how 3D structures can be reconstructed, refined, and validated, I'll touch briefly on some of the advanced refinement concepts that help us obtain higher resolutions in interesting cases. One area where several recent advancements have been made is in the correction of aberrations caused in the microscope during imaging. Several groups have shown that ever smaller imaging defects can be estimated and computationally corrected. Both stage drift and sample deformation occurring during imaging can be corrected by capturing movies rather than single frames in the electron microscope. The microscope's optical characteristics, parameterized by the contrast transfer function, CTF, can also be estimated locally at different positions within the sample and provides a way to deal with tilt, bending, and otherwise irregular samples. For the highest resolution results, it is critical to account for higher order aberrations as well, such as coma in the microscope. Today, in software tools like CryoSpark and using the latest GPUs, these steps can all be done automatically and extremely fast, faster than data collection itself, which is important as we will discuss later. Another direction in which developments have been made is in the creation of algorithms specifically designed for certain types of target molecules. Membrane proteins, which as Jakob mentioned, constitute about 30% of drug targets today, are special in that part of the protein is naturally embedded in a cell membrane and part is in the solvent. These proteins can have very different characteristics in different parts of the molecule, and when purified in a cryo-EM sample, they are partially enveloped in a disordered detergent micelle, which creates further intrastructure variability. By changing how structures are represented, CryoSpark's non-uniform refinement algorithm automatically treats different regions of the molecule optimally given their characteristics. This can, in many cases, yield dramatic improvements in map quality and interpretability for membrane proteins. Along with disorder, flexibility in protein molecules is very common. In fact, almost all proteins are molecular machines that have moving, bending, and rotating parts that work together to mediate a biological process in the cell. With the basic iterative refinement algorithm I mentioned earlier, flexibility is a severe limitation. Particle images of flexible molecules will not coherently average together at high resolutions. Instead, details are blurred out by the flexing of the molecule. Dealing with flexibility is an open area of research today. Current techniques include performing refinements of subvolumes, as shown in this image. Each subvolume is treated as a rigid structure and joints are discontinuous. There are also some diagnostics available to assess the presence of flexibility. As new developments, we are working on some tools to directly detect and at least visualize flexibility of molecules from the EM data itself. This video shows a simple version of that in 2 dimensions. On the left is an SNRNP protein, and looking closely at the 2D result, we can see that the entire protein bends and deforms. All of that motion is actually contained within the EM data itself, and with the right algorithms can be recovered, as you see. On the right is an ATPase molecule, where the bending of the central stalk causes the entire protein to wobble, and is likely a reason for limited resolutions in refining the structure. As you can see, CryoEM data processing is not a trivial workflow. Heterogeneity of more than one structure and state-of-the-art resolutions require more time and larger datasets. Multiple manually guided iterations through the processing pipeline are required for near-atomic resolutions and the identification of small details like ligand binding sites. Typical dataset sizes now easily reach several million particles. So along with building efficient and powerful algorithms to deal with this, a seamless software system is necessary to handle large volumes of data quickly, reduce wasted user time, and get reliable results. Towards that goal, we have designed the CryoSpark software system to be completely intuitive, visual, and experiment-based. It simplifies CryoEM data processing so that even complex workflows can be easily managed by all users in a repeatable and inspectable manner. A drag-and-drop interface, user and project management tools, and automation tools make CryoSpark very powerful for industry workflows. Along with software, hardware systems need to be designed to handle the specific requirements of CryoEM data processing, especially to achieve scalability. Several CryoEM programs, including CryoSpark, are GPU accelerated exclusively using NVIDIA GPUs. The raw parallel processing power of a GPU is far better per dollar or per watt than CPUs, and CryoEM algorithms nearly all have highly parallelizable core computations, which generally benefit greatly from GPU acceleration. It's also worth noting that usually the algorithms scale well with multiple GPUs available in a node. For those implementing systems for CryoEM computation, it's important to note that with the latest algorithms and GPUs, reading data from storage is often the first bottleneck. To achieve the fastest and highest performance results, especially in CryoSpark, it's crucial to couple GPUs with very fast SSD disks. It's also important to have a plan or strategy to deal with the vast total amount of data that is collected and processed for CryoEM. A microscope can generate multiple terabytes per day of raw data, and typically these need to be saved for at least until a project is complete and usually even archived afterward. Multi-user and multi-project scaling also generally requires a cluster setup with at least a fast shared file system and multiple worker nodes, each with multiple GPUs available. Now that we've covered the algorithmic and practical aspects of computing 3D structures, I'll hand it over to Jakob to go into more detail about how these tools and CryoEM are actually being used today within the drug discovery context.
Thank you, Ali. So as we have explained, simply determining the structure of proteins, targets of interest, helps to understand their mechanism of action and could be used to identify a binding pocket where a molecule could interact as a drug. Besides the examples I gave you before, several important targets that have been enabled for drug discovery by CryoEM in the last few years are, for example, the tau and beta amyloid fibers. Both types of fibers aggregate in the brain of Alzheimer's patients. Their structures were solved from the helical arrangements that they make, not from single particles, another technique used by CryoEM for proteins that arrange in that type of structures, the helices, in which we use different kinds of algorithms and equations to solve the structure. In the top image, you have two kinds of filaments in which the tau protein arranges, but by interacting through certain residues. Other types of morphologies are involved in other neurodegenerative diseases. The amyloid beta protein arranging a similar helical structure and was also solved by CryoEM. Another example, the TRBP1 channel was solved recently. This channel is involved in sensing and transducing temperature. It's an example of the family of TRBP channels, many of which have been studied by CryoEM, about 50 in the last few years. They are very difficult to work with because they express very low and their conformations are very sensitive to chemical and physical stimuli. Besides target identification, one of the most exciting advances that CryoEM offers is the possibility of solving the structure of different conformations for the same protein in a single preparation. The proteins can modify their structure during the execution of the function, and every different structure that the protein can adapt is called a conformation. If the number of conformations is discrete and statistically relevant, CryoEM can produce individual structures for them with a single data set from a single preparation, giving an important amount of information to understand the mechanism of action of the protein and then to be able to treat the disease. One example is the human protein ATM involved in DNA damage response. Its structure was solved in several different conformations from a single preparation using the CryoEM algorithm that allow automatic 3D classification. Many of the examples of structure-based drug design remain private in the domain of the pharmaceutical companies due to intellectual property restrictions. One of the public examples that we can review in the literature comes from the structure of the drug mefloquine, an antimalaric medicine in complex with the plasmodium ribosome. The structure of this complex was solved by CryoEM and has served to design derivatives with improved binding and better antiparasitic effect. In the figure in panel B, you have a representation of the drug in its binding pocket in the ribosome. The ribosome is in gray, showing the pocket where the compound in green fits. You can see how there is an empty pocket, the red circle, that is discovered thanks to the structure. A modification of the molecule by adding an aromatic ring that fills that empty pocket allows a more intimate molecular interaction between the protein and the drug, as you see in the panel C, improving the binding and showing a better antimalaric effect in the derivative. On the side of conformation analysis and heterogeneity classification, an important work published by my colleague Giovanna Scapin is the analysis of the structure of the insulin receptor. This protein arranges as dimers in the surface of the cell, and its dysfunction has been associated to many diseases, from diabetes to cancer and Alzheimer's. It has a portion that is exposed to the outside of the cell, called the ectodomain, and a portion that is inside the cell. When the insulin binds the ectodomain, it triggers a conformational change that sends a signal inside the cell, and you're going to see that in the next video. By CryoEM, Giovanna saw the structure of the ectodomain portion and was able to infer the mechanism of action that we can see in the video. In light blue and green, you have the two different molecules that compose the dimer. And there it comes, the insulin in blue, that first binds to one of the proteins, triggering a conformational change that propagates through the monomer where it binds, but also changes the other monomer and at the same time generates a binding site. And this state that you are going to see right now, the full conformational change, is one of the two states solved by CryoEM by Giovanna. These conformational changes propagate to the intercellular domain, activating the signaling pathway inside the cell. Then the binding of the second molecule of insulin that comes right now, and that happens with negative cooperativity in the full receptor, fully activates the dimer. This fully activated receptor, here it comes the insulin, and this structure that is symmetric was the second structure solved by Giovanna in the same preparation. Beyond the use of small molecules as drugs, pharmaceutical companies also use immunology and our own immune system through different ways to help us fighting the diseases. CryoEM can also be a great advantage for them by being able to study the interaction of proteins with therapeutic antibodies, what are called antibody-based drugs. An antibody is just another type of protein, and you can see an example in the image at the left, where the antibody is yellow and the target protein is green. And the antibody is just another type of protein that can interact with very high specificity with a protein target. So CryoEM can study interactions of the antibody with the target at atomic level to be able to modify it in the same way that we do with a small molecule by changing the amino acid sequence of the antibody, and therefore improving its specificity or affinity for a binding site and changing the properties of a drug, that is the antibody itself. Other two uses that I want to mention briefly are the possibility of, just by visualization and gold labeling, label molecules with gold to increase contrast and characterize biochemically or chemical mechanism. In the picture in the middle, the label of the individual units of a polymer allows to understand the results of different conditions applied to the polymerization reaction. So you see the black dots are the gold clusters that are labeling every one of the monomers of a polymer that you see that is branched. And by pure visualization also, we are able to study what is the effect of different formulations of drugs in liposomes or lipid nanoparticles. That's the image at the right. And adjust them properly based on the size, shape, or features, contrast, or diffraction if there is a crystalline state inside a liposome. And with this, I leave the rest of the talk about the coming advances in throughput of this technique to Ali.
Thanks, Jakob. With these fascinating examples in mind, we can look forward to some of the advancements that are going to make CryoEM even more effective in the drug discovery context. Advancements are currently happening on all fronts. In sample preparation, multiple teams are working on commercialized sample preparation machines and robots to provide repeatability and automation, as well as to improve the overall quality of prepared samples. In imaging, microscope cameras continue to improve, yielding higher signal-to-noise ratios and larger capture sizes and frame speeds. And some types of advanced optics, like faceplates, are under investigation. On the computational front, there are several areas of development, as we discussed. But one of the most important near-term goals for drug discovery is to minimize the time to getting a 3D structure once a sample is prepared. There are new algorithms to be developed for difficult targets, membrane proteins, antibody complexes, as Jakob mentioned, and data processing must be optimized in tandem with data collection. All of these advancements will continue to increase the utility of CryoEM as well as its ROI as an investment for the pharmaceutical industry. On that front, I'm very excited to share one of our latest developments. We've been working on a new CryoEM computing system, CryoSpark Live. CryoSpark Live is a fully interactive, real-time processing solution performing the full pipeline of CryoEM computation in real time, concurrently with data collection in the microscope. A well-tuned CryoEM microscope can output upwards of 5,000 images per day. CryoSpark Live can easily perform on-the-fly pre-processing of the raw data at double that rate on simple hardware. Large datasets can be filtered and curated as they're being collected, so that bad images, contaminants, and bad ice can be detected and discarded immediately. Processing parameters, particle picking tools, and diagnostics are all available interactively. And while on-the-fly pre-processing is helpful, the most important parts of CryoSpark Live are in providing real-time 2D and 3D reconstruction of protein particles. A new type of streaming 2D classification provides up-to-date results within just a few minutes of a new image being captured and allows for inspection and curation of particles on the fly. And most critically, CryoSpark Live provides for the first time real-time streaming 3D refinement, which also updates within minutes of new data being collected, giving the tightest possible feedback loop and fastest time to structure. The refined 3D structure gives the user a clear indication of the quality of data being collected in the microscope at the same time, and closes the feedback loop so that decisions about the data collection process itself can be made based on the actual 3D structures present in the data. Tools like this, along with the computational infrastructure that supports them, are steps towards the goal of CryoEM being a robust, scalable technique within drug discovery. With that, we hope everyone is as excited about the upcoming and ongoing developments in CryoEM as we are. It's certainly a very exciting time in the field. I think we're going to move on now to questions, so thank you all for listening to both me and Jakob. Thank you.
Thank you, Ali, and thank you, Jakob. This is John Leffman from NVIDIA. We are going to take some questions. So we have some questions that have been written in. Please continue to stream questions in. I think there's some interesting questions that have come up really about what are some general challenges that are in the field that we're trying to overcome right now. Ali? What are the next steps that are out there that are not being tackled right now?
Sure, that's a great question. I think looking forward into the future, sample preparation, as I mentioned on the last slide, is an area where there's a lot of room for improvement, and only really the first generation of automated sample preparation machines and robots have been developed and are currently being commercialized right now. So I think there's a lot more work to be done on that front, and the quality of a sample can dramatically change what resolutions you can expect from a data set. I think Jakob can probably talk more about this as at Merck, the hands-on experience of actually dealing with this issue. But sample preparation is an area where there's a lot of room for improvement, especially to deal with extremely small proteins, which are currently beyond the realm of what CryoEM can handle, but are interesting targets for drug discovery, as well as things like flexible molecules or molecules that have an affinity to the air-water interface, which often causes damage. On other fronts, on the computational side, I think I mentioned a lot of the forward-looking ideas. One of the notions that comes out of, for example, this idea of CryoSpark Live is being able to automatically determine how to collect data optimally in a microscope. I think that's a frontier that a lot of people are excited about, because once you can get 3D structures in real time with data collection, there are a lot of questions to be answered in the microscope itself. For example, when you're looking at a particular sample, a particular grid with certain holes, which holes and which squares should you actually pick for imaging? Which ice is actually going to yield good images? So there are lots of questions on that front. And I think flexibility, which is another one that I mentioned, there's a lot of room for improvement in dealing with flexible molecules, which, like I said, really are almost all the proteins out there, and especially for conformational changes that drug teams might be interested in. When a drug actually binds to a protein, it almost always causes a conformational change, as Jakob showed with the videos of the insulin receptor. And those conformational changes generally involve flexibility or flexing of the molecule itself to accept the ligand. So I think that's probably a good summary if Jakob wants to add anything.
Yeah, I think that you describe everything very well. I think that basically we need speed compared to other techniques. If we want to screen a few hundred targets, we need to do it as quickly as possible. And we need to the field is quickly overcoming limits in size. For CryoEM, the larger the protein, the better. But so the small proteins are quite difficult. Very small proteins are quite difficult. But you can see how the field is evolving, going to smaller and smaller targets. And basically, sample preparation to make the experiments more reproducible would be something that we would like to have more automation and to have a production. But I think that the field is evolving very quickly in all the aspects, right? And this is because it's right now very interesting for many laboratories around the world and pharmaceutical companies, and then there is a lot of interest in making things better. And things are happening basically every few months.
Thanks. What do we think about tomography, electron tomography for drug discovery?
So we are experimenting. I think the pharmaceutical companies are experimenting with tomography, but tomography responds to very specific questions. So in tomography, you want to visualize at the level of the cell or large structures in the cell. So you can use it to find what target where the target is, or what's the target that responds to your drug if you don't know it. But so I think it's pretty promising, and it's a field that is also evolving at the same time. But it's a different approximation. I don't know exactly the perspective of other companies. I think that everybody is exploring this possibility.
Thanks. So I think there's several questions that are relating to sample preparation and flexibility. I think that there are quite a few that we're not going to get to today, and we're going to try to have some follow-up answers. But I think that the overall questions are relating to quantifying sample preparation in terms of ice quality, purity of samples, and things like that that are sort of the pre-processing steps that go into it, even parts of the data collection process. Ali or Yakov, any comments there?
Yeah, so I think that sample preparation. Well, we have different things here, right? So to be able to express and purify a protein with quality and with enough quantity is a problem that extends beyond CryoEM. It's something that is related to science and structural determination. So we all need good quality proteins, and some specimens are really difficult to work with because of many reasons, and some reasons that are unknown. That's doing science, right? So that's one part. The departments that are producing protein, the people that specialize in express and purify protein, they have to fight different wars with every project to be able to give a quality sample. That's on one side. On the side of CryoEM, how you deal with this sample in the way you prepare it right now is quite manual, unfortunately, but we are in the process of automation, and in the field there has, in the last few years, a few robots have been created to reduce to the maximum the sample handling by the humans, and this is only because the size of the sample is so small, you use grids that are 3 millimeters that contains thousands of holes that are 1 micron, 2 micron size. So every minor change, even if you believe that you are doing everything the same way, you introduce a lot of variation in the samples, and that is bad for reproducibility and for production. So we are trying to reduce that. Well, the field is trying to reduce that as much as possible, and I think we are in the right way. This is obviously one of the things that we have to improve the next few years.
Okay, thank you very much, Jakob. Ali, quickly, do you have any comments to add on to the last question before we wrap up?
No, I think Jakob covered it very well.
Okay. Well, thank you so much. I want to thank our presenters, so Ali and Jakob, Structura, and Merck for presenting today. Excellent presentation, and there are plenty of questions to follow up on. So we'll do our very best to answer questions and follow-ups through sent out as an email response. And thank you to our attendees for coming to today's webinar. And we will close it down for now. Thank you. Have a good day.
Thank you. Have a good day.